Have you ever just wanted to get to the bottom line of something complex but like really fast?
Yeah.
Like how do you know if that new ingredient in your multivitamin, is going to give you cancer?
Oh, yeah.
Well, that's what we are talking about today, carcinogenicity testing. That's our deep dive today.
We're going to break down kind of how those potential cancer risks are evaluated from
Right.
all sorts of things like medications, chemicals, food additives, all that good stuff that you encounter on a daily basis.
And why should you care?
Well, because this is all a part of, preclinical safety assessments for all sorts of things. And this helps inform decisions that are made by regulatory agencies like the FDA and EMA, which you've probably heard of.
Right.
It's really all about protecting public health,
Huh.
identifying those substances that could increase your risk of cancer.
Okay, so how do scientists figure out if something causes cancer?
So the kind of gold standard right now is long-term animal studies. These are called in vivo bioassays.
In v.
In vivo, yeah. And they typically last about 2 years.
Wow.
Yeah, in rodents like rats and mice.
Huh.
And these studies are designed to really mimic potential human exposure over a lifespan. And they use rodents because there are physiological similarities between rodents and humans.
2 years is a long time. What are they looking for over those 2 years?
So researchers will give the animals the test substance, whatever that may be, at various doses, including the maximum tolerated dose, which they call the MTD.
Okay.
They also use routes that are relevant to human exposure, like eating it or breathing it in or putting it on their skin. And then they track the animals very carefully for tumor development, looking at the number of animals that develop tumors, how quickly the tumors appear, how many tumors each animal gets.
So they're basically seeing, does this group of animals that was given this substance develop more tumors than the control group?
Exactly. And then at the very end of the study, they do a microscopic examination of the tissues, which is called histopathology. And they do this to identify any cancerous or precancerous changes.
So the main goal is to look at tumor development.
Yeah. They also use statistical analysis to compare tumor rates between groups to determine if there's a significant increase.
So are animal studies the only thing they use?
No. There are, of course, advantages and disadvantages to animal studies. It can be really expensive, and obviously it takes a lot of time. There are ethical concerns, using animals. And of course, results in rodents don't always translate perfectly to humans.
Right.
But there are also other ways that they test for carcinogenicity. There are things called in vitro tests.
In vitro.
Yeah,
Oh,
in
wow.
vitro. So these are basically cell-based methods.
Okay.
And they're really good because they're a lot faster,
Okay.
and they can provide sort of an initial assessment.
Gotcha.
One example of an in vitro test is the Ames test.
Ames test.
Yeah, so the Ames test is a really common bacterial test. It's
Right.
really quick, it's inexpensive, and what they're really looking at is, can a chemical cause DNA mutations? So the rationale is that if a substance can damage bacterial DNA, then it might also be able to damage human DNA, which of course could lead to cancer.
Right.
But remember, this is just bacteria. It's not, a whole organism.
So the Ames test is sort of like a first pass.
Yeah, exactly. It's an early warning sign. It doesn't necessarily mean that it's going to, definitively cause cancer.
Yeah. So what else?
There are also mammalian cell transformation assays, which look at early cancerous changes in mammalian cells, like if the cell changes shape or if it grows uncontrollably.
Gotcha.
And so this is a mammalian system. It's not bacterial, but it's still just looking at cells in a dish.
So it's still kind of a simplified version.
Right, exactly. And then there are some other in vitro tests, like the chromosome aberration and micronucleus assays.
Wow.
Yeah.
Those are big words.
Big words, yeah. But basically, they're just looking at chromosome damage, which is, another indicator of genotoxicity, if something can damage your DNA.
So we've talked about animal studies. We've talked about in vitro. Are there any other ways that they test?
Well, there's so much computational power now.
Yeah.
So there are what are called in silico methods.
In silico.
Yeah, in silico. So these basically use chemical structures and existing data to predict carcinogenicity.
Oh, wow.
So basically, they use computers.
Okay.
So like QSR models are one example. They identify links between a chemical structure and its ability to cause cancer based on what's already known about similar chemicals. And then there are also machine learning and AI models.
Wow.
Yeah, they're really cool. They analyze large data sets to make predictions. So these in silico models are really good for initial screening
Okay.
and for prioritizing substances.
Hmm.
But, they're only as good as the data that they're trained on.
So how do they decide if something is a carcinogen or not with all these different
Yeah.
tests?
Yeah, so basically they look at all the evidence. A really strong indicator is a significant increase in tumors in animal studies, ideally in multiple species, in both males and females, and at multiple doses.
Okay.
And then they consider the results from the in vitro and in silico tests as well.
So kind of like everything together.
Exactly. And if they see a consistent pattern across all those different layers, then that suggests potential carcinogenicity. Sometimes, of course, the results are unclear, and then they would do more testing.
Right. So they start with like the Ames test and then they move to the animal studies if they
Yeah,
need
it's,
to.
it's a layered approach.
Oh, that's really interesting.
Yeah. The whole point is to protect your health.
Right.
And to evaluate the safety of all these everyday products.
Right. All the things that we use.
Exactly.
Well, it is fascinating to think about how much work goes into this,
Mm.
and the different approaches and the limitations of each approach.
Yeah, it's a complex issue.
It really is. And so I guess the question for our listeners is, what does this tell you about, the efforts that are ongoing to ensure the safety of the products that we use?
Mm-hmm.
Something to think about. Well, thank you so much for breaking all of this down. This was really interesting.
My pleasure. Thanks for having me.
And to our listeners, thanks for listening.